Oh, thank you. Can you hear me?
Yes.
Okay, so it's let me know if there are any problems with the screen sharing, and do stop me along the way if I like to be stopped and ask questions and so forth. So yeah, so I'm an associate prof at University of Pennsylvania and always looking for grad students and postdocs. So if you're interested for some time in the future, talk to me. And I'm presenting a work in progress. This hasn't been published, and as a couple of different, touching upon two different directions actually that I've been working on for a few years now with my students, Katie Kochatova and Jacob Stanger here at Penn, and with my long-term collaborator Tiho Kostadinov, who and a newer collaborator Siobhan Roy in the UK. These profs are developers of satellite-based algorithms, and my role in this is to understand their algorithms and compare it with other products such as observations and climate models and so forth. So I guess to start off, it's important to say that I mean, the questions I want to answer today are: how can we see phytoplankton carbon from space, and will phytoplankton modeled by the new generation climate models look anything like those retrieved from space? Okay, so now traditionally satellite retrieval has focused on chlorophyll. Okay, and chlorophyll in turn has been used to estimate phytoplankton carbon biomass and productivity. However, the ratio of chlorophyll to carbon biomass is known now to vary with light and nutrients, basically making the interpretation of chlorophyll products tricky. So a new generation of satellite algorithms over the past few years have popped up, focused directly on retrieving carbon biomass from space. Now, rather than simply chlorophyll. Okay. Now, there are only two of these algorithms at present that are specifically aimed at retrieving size fractionated carbon biomass. Okay, and those are the ones that I will be discussing today. Now, why size, right? Size of phytoplankton is important for ecological and biogeochemical reasons. For example, we know that larger phytoplankton, such as diatoms or coccolith, can result in stronger flux of particulate organic carbon to the subsurface, intensifying the biological carbon pump, right? Storing more carbon in the subsurface. We can say that to a first approximation, phytoplankton size groups, right, approximates phytoplankton, these phytoplankton functional group, right? So we're going to try to estimate size groups with the hope that this will tell us about the structure of the pelagic ecosystems and its role in the global carbon cycle. Now, the basis, so here are the algorithms that I'll be comparing, the Roy set of algorithms and the Kostadinov set of algorithms. And the basis behind both of these algorithms is a particle size distribution that is quite well known and well established in ecological systems. So if I plot the log of the number of particles versus the log of the particle size, what we know is that, and we've observed for decades now, is that there are always more always more small particles, right? So this is small on the left and this is large on the right. This is just an example from the Santa Barbara Channel. We see that there are always more particles that are small compared to particles that are large. And the theory behind this is we can spend days thinking about this, but the idea is that, well, excuse me here. Whoops. But the idea is that this relationship can be written as n, the number of particles, as a power law of the size of the particles d, where d naught and n naught are constants in this case, and the slope of this line c is called the PSD slope, or the particle size distribution slope. Okay. Now, the trick here is that this particle size distribution can be determined from either backscattering or light absorption. Okay, and why is that? Because the size of the particles determines how much a particle backscatters light coming in, or how much, or conversely, how much light it absorbs. So there's a similar relationship, if you can imagine in your head, between the amount of light backscattered and absorbed and the size of the particles and the particle diameters, and that is what actually satellite products get, right? So a relationship between backscattering and particle size. That relationship is retrieved, and that relationship actually is used to derive this PSD slope that we see here. Okay, now given that this PSD slope that's retrieved from the satellite products, okay, and with the assumption of this power law spectrum, we combine this relationship with what? Well, we also assume, sorry, this is the second assumption. So we assume a model, a power law model here. We assume also a relationship between biomass of carbon and the volume of the cell. Okay, so now it's not density inside cells is not always right. The biomass of the cell is not equally distributed in the cell, and it can get a little bit complicated. So we use lab measurements, so compilations. In our case, we use the compilation Mendendorf 2000, and to understand the relationship between the biomass and the volume of the cell. Okay, and then what we do next is we integrate over the biomass of the particles within a given size group. So now we have d mean and d max are size groups of and these size groups and size categories for us will be in the pico, 0.5 to 2 micrometer, nano, and micro range. So we assume that the particles are all spherical, and we integrate the biomass over all of these particles, assuming a simple relationship between the volume and the biomass. So now both of these algorithms for this exercise, we used 5 years of CWIF's data set in the 2000s, and both algorithms use the same procedure of calculating the total, of a very similar procedure of calculating the total biomass with similar coefficients. What differs between these two products is the particle size distribution slope. Okay, now the first product, which is the Roy product, is an absorption-based product. Okay, and here's a set of on the left-hand side. You can see the an absorption as a function of wavelengths. The Roy absorption, Roy uses absorption at 676 nanometers, which which basically means that this is the peak of the absorption for phytoplankton. So this product is sensitive to phytoplankton only. For example, detritus, which we see in the red line, is not very present in this. Dead organic matter is not very present here. Okay. Now, if we go instead to and color dissolved organic matter, doesn't have much absorption there. Now, the Kostadinov data set uses a combination of multiple backscatterings at 410, 490, 550, 4 backscattering wavelength, and because there is backscattering for principally of detritus, as we see here. So the detritus is pretty dead organic matter. What we're not showing here are zooplankton. It is also possible for zooplankton to scatter in this wavelength. So we established that the Kostadinov data set will represent primarily not just phytoplankton, but it will also include perhaps detritus and zooplankton. So we call this a POC product, particulate organic carbon product. Okay. So here the size ranges we're using the same size ranges for all of these, for both of these products. And the idea is that within the size ranges, pico, nano, micro, right? We have zooplankton, organic detritus, bacteria, depending on the size range. And the idea here is to look at the particulate organic carbon as being the sum of phytoplankton and something else, which in our case is some non-algal particles, right? So organic matter, maybe organic detritus, or maybe zooplankton, and so forth. So the question is, can we use these two together to tell us something about this non-algal particles? But because very, very little is known about these non-algal particles, and there's extremely little literature on these. So many different questions. So we can go here, and I'm just going to show a couple of slides from this project where we can understand the global patterns of size fractionated phytoplankton, particulate organic carbon, and also hopefully non-algal particles, right? Get to non-algal particles. Now, as I was mentioning, these two products, the Kostadinov and the Roy, what they do is they retrieve the particular size PSD slope, particle size distribution slope. Here's the number of particles, well, log, I should say, versus the log in the size of particles. And what's interesting is that the retrieved slopes at each point, right, show us the relationship between the number of particles and the size of particles at each point in the ocean. Okay, this is a calculation done for the same 5 years in the two products. And in both products, we see that the slope, right, this C coefficient, which is the slope here, is larger. The red numbers is larger in the subtropics compared to the high latitudes. So what does this mean, right? Well, because small phytoplankton, right, the pico phytoplankton, have in general affinity for low nutrient environments, we will find a lot of small phytoplankton in the oligotrophic gyres where we have very few nutrients, right? In contrast, nano and micro phytoplankton, the bigger guys, right, do much better in high nutrient upwelling zones and high latitudes, right, where they can take advantage of nutrient pulses. Okay, so that's why the slopes are different, right, in the high latitudes and in the subtropics. Now, these slopes in the PSD slope are get translated in these two codes in the total biomass in each, and the biomass of phytoplankton and particulate organic carbon in each of the size groups. And here I'm showing for you how much biomass, the relative amount of biomass, phytoplankton biomass and particulate organic carbon biomass, the percent of in each phytoplankton, in each group, size group. So again, this is just to confirm that the percent of the smallest guys, the pico, the picos live primarily in the low latitude oligotrophic gyre, as expected from previous work. And while the larger guys, the nanos and the micros, do much better in upwelling regimes on the west, for example, on the west coast of the Americas and Africa, and the high latitude subpolar regimes. Okay, so now So that's pretty, and we do a first order, right? The patterns of on the left-hand side and on the right-hand side look similar, and we would like to, as a next step, we also want to understand the differences between these products, because the differences can tell us about these completely unknown detritus and zooplankton and other beautiful things that we've never known about before. Now, another way to question this data set is to look at the total amount of biomass. These are the phytoplankton total biomass and the POC total biomass from the Kostadinov and the Roy data sets, and we can look at the ratio of these.
Can I just stop you for one second? Oh, yes, perfect. You fixed it. I was going to ask you to move the, yeah, that's perfect. No problem.
Okay.
You couldn't see the figure.
So basically, oh, you don't see it if I move? Okay.
No, it's fine. It was just cut in half, the rightmost figure, but it's you fixed it. Perfect.
So the interesting thing here is that based on these two products, what we get is that the if you look at this, is the ratio, and these numbers are very small, right? So what percent of this is the ratio of phytoplankton divided by particulate organic carbon. So traditionally, so people like Mike Barenfeld and so forth assumed a ratio of 3. So he assume that phytoplankton are just 1/3 of the total particulate organic carbon. Others have come after that and have said, well, but maybe it's not a uniform ratio, and maybe this number is not 3, but it's closer to 5. So there's 5 times more stuff in POC compared to phytoplankton. And recently people have started to think, okay, how does this ratio vary spatially? This is not a very good color scheme, but basically we're finding numbers that are lower than what other people have found. So the phytoplankton biomass, I'm only finding it to be, 5% to at most 1/5 of the total POC biomass. And we really have no idea what this number should be like. There, just to say that importantly, there are a couple of other, so a few other particulate organic carbon products. And one is a classical Stromsky POC, and another is a classical Barenfeld particulate organic carbon. And the Barenfeld gives very different values compared to the POC that we have here, while the Stromsky is pretty similar. So this ratio varies very much depending on the product that you use, okay? Now, to say a few words about, to go into the physics a little bit of the system, one interesting and fun exercise we can always do is to take the phytoplankton biomass. In this case, I'm looking at the middle group, the nano, which dominates in terms of biomass the ocean. So there are a lot of small phytoplankton picos in the ocean, but they are very light. So most of the biomass is actually in the second group, in the middle-sized group. And I'm showing here correlations between the phytoplankton biomass derived for this monthly biomass derived from CWIF's satellite NASA data sets for a 5-year period, correlations with wind speed, surface light, and surface SSTs. And what we see here is the typical patterns that we expect in the subtropics. If we have a period or a season, and that happens to be usually in winter time with more mixing, then we have more turbulence, we have more vertical mixing of nutrients from below, more nutrients at the surface, and more phytoplankton growth, where this phytoplankton growth is principally in the larger phytoplankton groups as opposed to the pico size groups, and that results in a growth in biomass overall. And the opposite, so that's those are the red lines. And then the opposite happens in the higher latitudes, where more mixing results in a deeper mixed layer depth and more light limitation. And more light limitation means less phytoplankton growth and ultimately less phytoplankton biomass. So there's some very interesting dynamics that confirms our first order intuition, and we're working on I'm working now to understand differences between the phytoplankton biomass and the POC biomass, and whether that can tell us again something about the seasonal dynamics of the detrital matter, which nobody knows anything about. Okay, so now let me spend 5 minutes or at most 10 on comparing satellite products with the outputs from the latest generation climate models. So a few words about climate models. There's a growing number of CMIP of Earth system models. Contributions in red are the CMIP 6 latest generation contributions, compared to in green the CMIP 3, in 5 the CMIP 5. So you can see that going from green to orange to dark red, there are more and more models in the red category, meaning that as we go from one generation of model to another, there's more and more excitement about climate models, more consortia being built, more of these climate models are come online. So a subset of these models contain ocean biogeochemical modules, not all. There are more than 50 climate models out there that participate, and a subset of these have ocean biogeochemical modules, and these biogeochemical modules can be of varying diversity. So you can have an NPZD model, then you can have a low complexity and intermediate complexity models with more and more sophisticated elements, and you can have high complexity models. Okay, so in my work, what I do is I look at, so here are some simple CMIP 6 models, and I take a bunch of these models that have relatively higher complexity because I'm looking to understand the phytoplankton. So among those high complexity models, for example, are the GFDL models, which have multiple phyto groups and zoo size groups, and the IPSL French model, and the UK, of course, model, and the CSM NCAR model. And these have typically at least 2-3 phytoplankton groups and at least 2-3 zooplankton groups. But we can also look at other models that are slightly simpler, and then compare across these models the behavior of the phytoplankton behavior. So here's, for example, the model biomass across for a 10-year modern period across I'm missing the units, across a bunch of these models, and immediately you see that the expected patterns from observations that I showed you is that we expect higher biomass in high latitudes and upwelling regimes, and lower in oligotrophic gyres, and that seems to be overall the case. But they're clearly, right, here's the German model, the MPI Max Planck Institute model, and they're clearly very big differences. And here's the UK model as opposed to NASA GIS or CNRM, or there are very big differences across these models. And these differences among the biological outputs tend to be much higher, and I've noticed this in the previous generation model, CMIP 5. Differences among biological products tend to be much larger than differences across models for physical products such as SST. So if I showed you the SST product, it would be very similar across these models. Biology is all over the place, okay? So one way to compare apples and pears, meaning comparing satellite algorithms on top and climate models on the bottom, is, for example, to try to look at seasonality. Right. So here's an index for a period that shows how much variability for a given can be explained by the seasonal cycle, as opposed to interannual variability or one-time events, or right. So how seasonal this product is, okay, and then we can do an averaging over 10 phytoplankton, over 10 algorithms, and so here are 10 different algorithms where we look at the seasonality of phytoplankton biomass, and on the bottom we look at a bunch of CMIP climate models. And what we see in general, right, is that the climate models are way more seasonal than the satellite algorithms. There's much more that we can use climate models. Well, climate models use, right, known equations, right? And the way we build these models, basically we put in, what we put in is what we get out. We make these models somehow to be way more seasonal, at least in high latitudes, than what the satellite algorithms, tell us. So it's also smoother spatial variability, while we look at the phytoplankton, at the satellite algorithms, and we see these patches of high seasonality that tend to be in the seasonally stratified subtropical gyres, at the edges of the gyres, which tend to correspond to sharp transitions of surface chlorophylls and sharp temperature transitions, etc. So the frontal structure doesn't seem to be well represented in the climate models. And that's the case. If I showed you 15 different CMIP6 models, it would be the same thing. They all tend to have the same patterns. So basically, we can't do fronts yet in the models, and we make high latitude dynamics be too easy, too seasonal, way more seasonal than we think it should be. So now,
can you move the slides a bit to the left again?
Oh my God. Okay.
They seem to be moving around. Oh yeah, that's better. Thank you.
So just to ensure that I go completely over time here, let me talk to you about one other little story here. And the classical story, for example, is that there are big differences in physics and therefore in ecology between the subpolar North Pacific and subpolar North Atlantic. This is Ocean Station Papa, which is a high nutrient, low chlorophyll environment, an HMLC region. And then this is a region along Nabe in the North Atlantic, where we are supposed to have tons of iron. Phytoplankton here is nitrate limited with large spring blooms, as shown here for the chlorophyll, right? While at Ocean Station Papa in the Pacific, what we have is because of the traditional theory, the classical story says that because phytoplankton don't have enough iron and they're grazed very aggressively, there's not such a strong peak at Ocean Station Papa as in the North Atlantic in biomass. So this is what the chlorophyll tells us. So then I ask, okay, I looked at a bunch of satellite products and a bunch of models to see, okay, can we see this difference across satellite products and across models? And the answer is, well, depends what products you look at. So here's the Kostadinov product, one of the two products I showed you today. Here's another classical NASA product, the Stramsky particular POC, and they look pretty similar to the chlorophyll, right? There's a huge spring bloom in the North Atlantic and not much bloominess blooming at the Ocean Station Papa. However, Westbury 2016 paper claimed that they actually see a very big bloom at Ocean Station Papa, and this bloom is in the summertime, which is a very different picture from what these other products have told us. So now, out of curiosity, here is the spread across a bunch of CMIP6 latest generation models, biomass at the Ocean Station Papa in the North Pacific at present. Here's biomass over the North Atlantic at present. And I just wanted to put these to show you, to shock you a little bit and to show you how big the range is, okay, across the climate model. So we're going from whatever the units are here, from 1020 to 175, right? So we have an order of magnitude variation among the different models in biomass at both locations. And it's not super clear that the biomass in the North Atlantic peaks any differently or at different times. It takes a lot of digging to try to understand, right, the dynamics of these processes. And here's the delta biomass, the expected change over 100 years in one of the RCP scenarios, the RCP 8.5 scenario. So you see that in some models, biomass at Ocean Station Papa is expected to increase. There are numbers that are positive and numbers that are negative. So we don't know anything about anything, it looks like, right? Do we know why should some models predict increases and some models predict decreases? Well, it depends on what your nutrients and your iron are doing, okay? So here's at Ocean Station Papa, we have nitrate across many of the CIMIP models. And in gray, I put observations from the World Ocean Atlas. This is nitrate in the North Pacific. Here I have surface iron at Ocean Station Papa. And sorry for shifting on you. In blue, I don't know how to move this. Oh, there you go. And in blue, I have observations and all of these models, basically, with one model exception, CNRM, with one model exception. Maybe IPSL doesn't do too bad. So with maybe two model exceptions, all the other 10 models that I have here have too much iron at Ocean Station Papa. If you have too much iron in the models compared to what you should have at the surface, that means that the phytoplankton are super happy. They bloom, especially the large phytoplankton and diatoms and so forth. And any phytoplankton that needs iron basically to grow, they're super happy. And then that means that they are going to eat up all the available nutrient. So in accordance with this, the nutrients in these models are going to be too low compared to observation. So we see indeed that in most of these models during the summer, the nutrients go to zero. So what's the conclusion here, or I call it anti-conclusions, is that just for this little story, right, that some satellite algorithms contradict each other, and some of the climate models contradict each other, okay? We see a pronounced phytoplankton summer bloom in most models, but only one satellite product, while the other satellite products show us spring blooms, okay? And that we see, for example, that all the CIMIP6 models, and I have the same thing in the CIMIP5 models, they tend to have way too much iron, and therefore the phytoplankton biomass is too large at Ocean Station Papa. And therefore, we cannot see this the expected observed big difference between Ocean Station Papa and Nabe. We can't quite see it in the models because Ocean Station Papa in the model seems to behave very similar to the North Atlantic. So models need to fix their iron and iron supply in order to get this right. So we're not quite at the stage where we can compare Earth system models with satellites at individual locations. I guess that's part of my conclusion. Most papers out there that try to combine, and there are very few actually, that try to match, satellite and climate models, they integrate across larger biomes to give more solid results, right? In general, the larger the areas you integrate over, the more stories you can see, right? So there's tons of work to be done to even be able to put on the same page in a paper, right, satellites and models. This also is a kind of cry for attention for people who use, and groups that actually use satellite models for data, satellite model output of chlorophyll and biomass for data assimilation. I've seen this done over and over again, and I know that many of the big groups do it, and I'm completely puzzled by it. I don't think we're at the stage where we can assimilate biomass and phytoplankton size groups and so forth in fisheries models, and because we have no idea what those products mean. There are 10 different products, satellite products, and each of them tells us a very different story. And then in the models, we have issues, big issues, I think, with iron specifically. So I'll just stop there. Tons of work to be done. If you feel you want to talk more about it, contact me. And yeah, so I'll stop here, just kind of throwing at you some anti-conclusions here.
Thank you very